We are not able to prevent or cure Parkinson's disease, in part because we don't know why neurons die in specific areas of the brain, such as in the substantia nigra. Several hypotheses have been proposed, but none of them has been confirmed. By the end of 2020, we published a paper in Brain where we show in human postmortem tissue that CD8 T cells infiltrate the substantia nigra in premotor early stages of Parkinson's disease, as well as in already diagnosed cases. Some of these T cells were actually contacting dopaminergic neurons. The highest infiltration of CD8 T cells were found in these early stages before Lewy body pathology and any kind of alpha-synuclein aggregation takes place, and more importantly, before dopaminergic cell loss can be detected. In addition, we found a positive correlation between the density of cytotoxic T cells and the rate of neuronal cell loss in already diagnosed cases. Altogether suggests that cytotoxic attack mediated by CD8 T cells initiates and propagates neuronal loss in Parkinson's disease. Our results demonstrate that the adaptive immune system is extremely relevant for the pathogenesis of the disease, and that the Lewy body formation, which is a hallmark of the disease, is preceded by CD8 T cells. Now, what we want to do is to understand why these CD8 T cells infiltrate the brain parenchyma and how they are linked to alpha-synuclein aggregation. And also, we will further characterize these lymphocytes. And last but not the least, we will try to determine the antigens that are recruiting these T cells to determine the etiology of the disease. This will make possible the establishment of preventive, therapeutic, and diagnostic strategies.